Hello, everyone. I see that we have a sizable number of participants for our AI interest group. As we discussed this spring, we will start in fall now a series of webinars, which will be a mix of our own people within the department and people from the outside, so that we get some ideas about what artificial intelligence can do in the field of environmental health and engineering, and also start to connect each other here within the department in order to enable projects, joint applications, and whatever. It's my pleasure to introduce today one of my team members, Alexandra Maertens. She's one of the resident bioinformaticians, data jugglers, and she has brought a lot of machine learning to our own group. She's going to talk about how to use AI for novel hypothesis generation. I don't want to take too much of your time, Alex. The idea is that we have about 30 minutes for this presentation, about 15 minutes for Q&A, and we will then use the remaining 15 minutes of the hour to discuss things relevant to the interest group in order to move forward. We have some guests which have not yet registered for our AI interest group, and I would very much encourage you to do so. It's an open process. I would say not even necessarily you have to be part of our department, but if you like to hear the discussions, trying to find our spot in this field, you're more than welcome to join us. Just send an email to Camilla or myself in order to get this done. And with this, Alex, please, it's your stage. I'm looking forward to hearing from you.
Okay, well, thank you, everybody. I'm here to talk about using AI for novel hypothesis generation. So can we stop looking under the lamppost for the lost keys? If you're expecting sort of a talk that goes knee-deep into algorithms, this talk is not for you. I want to talk more generally about how in toxicology we've kind of been looking at the same hypotheses over and over again, and why we might want to rethink this, especially when it comes to our understanding of genes and their functions. But to start off, I want to kind of think about one of the ways toxicology has been subject to what I would call big data of the wrong kind. And the best example of this is really bisphenol A. The bisphenol A has, oh, hang on. You want my video on? Camille, I can't turn my video on because it says the host has stopped it. So you have to take care of that.
Well, that's interesting. Keep going. I'll try to do that.
Yeah, I brushed my hair this morning, so it's not deliberate on my part. Anyway, so BPA has been subjected to multiple guideline studies in different species and different strains. The comparative toxicogenomics database shows that it's affects almost 21,000 genes, and that's 6,000 when you just restrict it to humans. So there's a lot of data on it, but the data isn't necessarily that informative. HSDB alone, the Hazardous Substance Data Bank, has 79 laboratory animal studies on this, and the recently completed FDA Clarity study involved 3,500 animals. But despite all this, there really isn't any kind of consensus about what really is a safe dose of BPA, and it's still being fought in the literature. And if you look at the literature, it's really extensive. PubMed returned over 10,000 abstracts for bisphenol A, and it's actually increasing at a rate of about 1,000 a year. And approximately 6,000 of those abstracts mention bisphenol A in the context of estrogen. And I got interested in this when we started working on the Mapping the Human Toxome project, when we were looking at estrogen genes, and then I sort of applied the same tools that we used to look at bisphenol A and see if it was the same genes or not. And when we did that, what we found was that if you use a very large transcriptomic data set with both estrogen and BPA, we demonstrated that yes, there were some similarities between high dose bisphenol A and estrogen, but there was really minimal conservation of the gene signatures. In other words, it was similar enough that you might think that they were the same mechanism, but when you looked at it very closely, and especially at the transcription factors, the estrogen receptor was not actually the key component. And in fact, low-dose bisphenol A in particular had a distinct set of transcription factors that were completely unrelated to the estrogen receptor, and the overwhelming majority of these transcription factors were very poorly studied. So this is an instance where people have generated an enormous amount of data about bisphenol A and its effects on estrogen and estrogen signaling, but they weren't necessarily looking in the right place. In other words, they were looking for known biology because that seemed like the obvious way to go, but they missed what was really important. And a lot of binding studies have shown that bisphenol A binds very, very weakly to estrogen, the estrogen receptor. And most unfortunately, for the things that it does bind to, a lot of the replacements for bisphenol A actually bind to that even worse. So the fact that we didn't really understand the biology has resulted in an unsafe substitution for bisphenol A. But one of the things I noticed when I was doing this is that when I looked at genes that were associated with bisphenol A, a lot of them were called orphan genes. So they were considered open reading frames. And this is sort of narrowly defined as genes without detectable homologs and other lineages, so we really don't know what they're doing. But fundamentally, they represent sort of dark matter, things in the genome that we really don't know what they do. We can't place them on any map. And this is really important for toxicology as well as just for the biomedical sciences in general, because every gene that we don't know is a potential target of a toxicant, and we can't really figure that out if we don't know enough about it to test. So I started thinking, well, you know, if we have all of these genes just in this one sample that I did that we don't know, how common is this? Or more broadly speaking, if you think of our map of the human genome, after all of this time, people kind of think that our map is closer to Google Maps. We know where we are, we know where we're going, we have it all mapped out. And the more I looked, the more it looked to me like the map was a lot closer to the medieval map of the world, where we had vast areas that were unknown, that for all practical purposes might as well be labeled "here be dragons." So I started looking at TCGA data. And in case people don't know, this was this enormous effort on the part of the cancer gene of an NCI to develop this huge data set. So it's 2.5 petabytes of data. Which is just an astounding amount. There's 33 different tumor types, 10 were very, very rare cancers, 11,000 patients, and 7 different data types. And this data has done exactly what big data should do. It was worth more than the sum of its parts. It allowed us to see DNA alterations, mutations, and pathways that we didn't know exist. In some instances, we discovered new types or subtypes of cancer that people didn't know about. In the case of stomach cancer, in the case of breast cancer, etc. So it's truly been transformative in a way that I don't think anybody could imagine before this data was put together. So I decided I would use this data set to start looking more specifically at orphan genes, genes that we didn't understand, and see why, if there was any particular reason why they were unknown. So at first I just did a very, very simple analysis. I pulled all the genes that were prognostic for survival with a p-value of less than 0.001. And so this gave me about 6,000 genes that were associated with a poor survival in cancer. And of those, there were about 115 that were open reading frames. So they were, you know, again, not well characterized. And not surprisingly, there was almost no literature in PubMed on these genes. But what was actually really surprising was that this was also true of most genes. So I sort of had assumed that this is cancer. This is, you know, one of the best studied diseases that we have. It's had all these huge data sets available for a long time. Surely we know a lot about the genes that are prognostic for survival in cancer. But as it turned out, over 4,000 of those genes that were prognostic for cancer had less than 50 PubMed IDs. And 2,000 of them do not show up on any pathway database. So they're going to be completely invisible in many bioinformatics workflows that depend on pathways. So when you started looking more broadly at the biomedical literature, you find that it's focused on a handful of genes. So there's a few genes that would have 6,000, 7,000 publications. Those are sort of the usual suspects like TP53, et cetera. But the vast majority of genes have very little biomedical literature. And separate studies have shown that this inequality worsens over time. In other words, the genes that already have a lot of literature tend to get more literature over time. So this is another example, I guess, of the rich getting richer. So we decided that we were going to use the TCGA, and we were going to kind of define this sort of arbitrary cutoff of 50 PubMed publications and describe that as the dark matter of the genome. So these were the genes that had not enough literature to really classify them and talk about them. And so the first question we asked was, well, why are these genes missing from our map? And if you looked at it at a very basic level, there were a couple of things that stood out. For one thing, these genes that had a poor literature base tended to not have a Mendelian inheritance pattern. So they were much less likely to have either autosomal recessive or dominant inheritance compared to the better studied genes. And this sounds like kind of a trivial thing, but when you think about it, one of the main ways people do, for example, dimensionality reduction in GWAS studies is to use pathway-based approaches. But if a lot of these genes that are likely involved in the sort of things that GWAS is looking at aren't on pathways, we're going to kind of be walking in circles a bit. So the fact that this literature is very much biased towards genes that show a very clear phenotype is going to start causing us problems later on as we try to use GWAS to fish out the sort of weaker genetic effects for things like diabetes, but especially the genes that are likely to cause some sort of predisposition to or sensitivity to environmental exposures. And not surprisingly, the well-studied genes were much more likely to be conserved in eukaryotes, and they were a lot less likely to be primate-specific. So again, this is not surprising when you consider all the data that's been dedicated to yeast studies. It's just easier to fish out genes that are highly conserved. But the other thing we wondered is, well, is it possible that these genes just aren't important? So the first thing we said was, well, all right, if we look at the correlation with the genes and the significance with any disease, to see if there was any sort of correlation, and the answer was just no. So we looked at, was there a correlation between the number of papers dedicated to a gene in Gleason score in prostate cancer, aggressiveness in colon cancer, survival in glioma? Nothing. So there was no indication just based on its significance to the disease that we were focusing our literature in the right place. The next thing we did is use WGCNA, and this is a network topology-based approach. This is the same approach I use for bisphenol A that is completely unsupervised to look at gene significance and potential interactions and functions. And this is what is really important. A lot of bioinformatics workflows look for enrichment of, a cell cycle pathway or DNA damage pathway, and usually that works. If you're looking for known biology, using a pathway-based approach will show it to you. But what it does not show you is all those other genes, the function of which we don't know. And so that is why it was absolutely critical that we use something that was unsupervised. So when we did this, we found that by any metric that you could use for a network topology-based approach, there was also no significant correlation between the number of publications and any network biology metric of gene importance. So again, there's a lot of different ways you can decide whether a gene is important or not, but there wasn't any indication that we were focusing our literature in the right place. So the next question we ask is, well, are there just areas of the map that were systematically overlooked? And this is really kind of critical because the genes that are missing from our pathway-based approaches are not missing at random. So when we started to look at the percentage of genes that were not studied in the various different modules that our unsupervised approach showed. For cell cycle, for example, 70% of the genes were well studied. But this is still kind of important to remember that even in the best characterized approach, there were still 35% genes that just didn't have a much literature base. On the other hand, when you looked at things like RNA splicing or mRNA processing, in those instances, up to 80% of the genes were not on pathways or lacked any literature approach. So it's important then to remember that some areas of biology are going to be easier to see based on what we already know, and some areas of biology are going to be much harder. There's just not as much literature. And most important, the information that you're missing is not missing at random. So to dive down, we started to think about, well, what types of genes are missing? So for example, apolipoprotein 6 was the highest ranking gene that was associated with aggressiveness in colon cancer. But when we looked at it on our unsupervised approach, it was actually highly correlated with STAT1 interferon pathway, and that was true in colon cancer, but it was also true in every cancer data set in TCGA. So that this wasn't just an accidental correlation in a data set, it was pretty consistent. And this would be completely invisible in a rodent model or a pathway-based approach. So it is annotated to a pathway. This is an example of a gene that did have some pathway annotations, and it was correct, but it misses the larger context of the interferon signaling. And although there are orthologs, these orthologs are actually not telling you anything important, because the correlation between the human and the rodent gene expression was essentially nil. So this is an example where the animal model of the gene or the ortholog exists, but it's uninformative, which is probably worse than no information at all. But it's also kind of interesting because understanding the immune role of APOL6 is pretty timely, because in a project we're working on COVID data sets, it actually shows up all the time, which kind of confirms that it was involved in interferon signaling, and also that it's probably important to know what it does. One of the other things we found was that there were a lot of non-coding RNA that were primate specific. So for example, C6or48 was the highest scoring primate specific gene in colon cancer aggressiveness. And it was also associated with lymphatic invasion of other cancers. When we use our unsupervised network approach, this showed a conserved interaction between two antisense RNAs, as well as genes involved in protein targeting to the ER and the RNA processing proteins. And this would have been completely invisible in a rodent model or a pathway-based approach, simply because the analogs don't exist. Another interesting thing we found was that there was a whole module in the glioma data set that had no statistically significant pathway interactions, or sorry, pathway annotations, or very few known interactions of the genes themselves. So in a lot of bioinformatics workflows, these sets of genes would just be completely ignored because there wasn't any reason to think that they were actually clustering together aside from an artifact of the data. So, you know, a lot of times people will just move along and look at other biology. But in this case, we decided to dig a little deeper, and we used a large-scale data mining approach to predict the functions of the genes, as well as text mining for the non-coding RNA. And this indicated that these genes were involved in central nervous system myelination and fatty acid metabolism, most of which was coordinated by a primate-specific non-coding RNA, C10 or 75. So again, this is kind of another instance of something that we were sort of be able to put a little bit tentatively on the map, even though you have to be kind of cautious when you're deriving stuff from cancer data sets. So one of the things that we want to do is to use this data to try to go forward and sort of build a better map. So one of the goals that we're working on now is to start identifying, cataloging the clinically important genes that have not been studied. And we especially want to focus on genes that would be invisible in non-primate model organisms. The other thing that we want to do is extend this by using a network-based approach to identify genes that are conserved. So they do have orthologs, but they have different functions in different species. And our preliminary data, just based on looking at the COVID data set, indicates that this is actually a pretty substantial function of genes. About 25 to 30% will just have very different functions in different species. The other thing that we're hoping to do is to build sort of a web-based tool that will allow researchers to quickly see if their genes, the genes of interest, act differently or similar in common animal models. One of the things that annoyed me a lot when we were writing grants is people would, often say, well, there's no animal model. And I always wondered, well, how do you know that the animal model is actually relevant? Sometimes it is, sometimes it isn't. And at least people have a way to kind of go forward and use bioinformatics to make a more educated decision about whether an animal model is really appropriate. So I hope that this has convinced you that we need to sort of embrace large-scale hypothesis-free data collection. In the traditional example of science, most people have really assumed that you start with some evidence, you build your hypothesis, and you go and you collect the data. But I think we need to start rethinking that and start imagining a world where we just start using a lot of data to build hypotheses and interpret that in the context of our evidence, but start looking more broadly and look at the data with fresh eyes. And I think one of the things that it is especially important to keep in mind is that one single well-done study of 1,000 samples is actually much more useful and much cheaper than 100 studies of 10 samples that are done inconsistently and under varying conditions. In other words, if you want your data to be worth more than the sum of its parts, you have to think about how you're gathering it. And to look at that in particular, again, I just pulled this from the comparative toxicogenomics data set. This is all the curated genes. So I restricted it to the relatively well-characterized genes that have been shown to interact with bisphenol A compared with estrogen. And there's a couple of things to think about here. First, you'd have to, for this to be true, you'd have to imagine that somehow bisphenol A is, interacting with almost 6 times as many genes as estrogen, which is a little bit, suspicious. You wonder if there's perhaps some publication bias. But even more specific, you have to kind of imagine that the overwhelming majority of those studies, over 90%, are actually based on one single reference. So in other words, there's 20,000 odd studies all showing that bisphenol A interacts with this gene and that gene. But this is an example where the data is actually worth less than the sum of its parts. Collectively, there's very little that you can do to look at the structure and the meaning of this. That having been said, you have to kind of be realistic about the challenges of working with that kind of data, because that data will always have artifacts. So this is a very famous example of the hazards of looking at big data. This was an fMRI study that was done on an Atlantic salmon to see if you were speaking to the salmon, what areas of the salmon brain were highlighted with this. And of course, the joke is, A, the Atlantic salmon are not considered to have great speech processing capabilities, but also the salmon in question was dead. So this was an example to show that the commonly used approaches to fMRI tended to produce a lot of artifacts. And in this case, it was just an example of failure to really correct for multiple comparisons. But this is an important point for toxicology, because the more we have such data, the more we'll have artifacts. And those artifacts, if they're out in the literature and become out as publicly available facts, can start to cause the same sort of problems that the misinformation about bisphenol A's mechanism has caused. And I will say that we kind of need to think about how we present our data. Usually when I, publish a paper on using WGCNA, I always add a lot of caveats that, well, this was based on a high throughput data set. There's always, correlations that are there by accident. And we usually get pushback from reviewers saying, well, how can you publish something, that you don't think is true? And the answer is, this is what hypothesis generation looks like. And this is a valuable aspect of science, as well as just going in with your predetermined hypotheses. Moving forward, to sort of get out of this impasse, I think we need to start thinking a little bit about what we want the long-term way we look at biology to be. So, if we start with all of this data that we gather from a molecular system, what we've been able to do so far is to use bioinformatics, to use information to connect that map together a bit and give it a little bit of pattern, a little bit of information. But when you think about it, that's not really very much. There's so much more that we could be doing. One of the things that we're working on in our lab is using the TCGA data and other data as well to start putting that data together in a way that is more focused on concrete mechanisms. So we want to use that systems biology approach to both build a model that you can then use for simulations. And then once you sort of understand your simulation better, you can start using that to explain how the system is actually working. Is there a feedback loop? Is it bistable? And that is what it truly means to be able to understand the system enough that we can start saying to people, well, this is a safe dose, this is not a safe dose. This is going to perturb the system too much. This is going to not perturb the system in a way that it can't recover from. And that's pretty daunting when you consider that we're still at the phase where we're trying to just use what information we have to sort of piece together our data in very basic maps. But ultimately, the more we do this and the more we're able to kind of fill in these blanks and develop better models, the more we'll be able to use that data to improve the maps and push what we know into the area of what we don't know. And that is it. With that, I will take any questions.
Excellent, Alex. Thank you very much for sharing this with us. I think you're really showing us how the future looks like, virtual experiments, simulation, and coming to the why in AI. So why are these data giving us these type of results? That's very nice. So I hope that we can now have everybody join us here, showing yourself. I think you should stop sharing, that we have a bit of a discussion. And I asked Camilla to, if possible, have everybody join in.
Yes, so this is set up as a webinar, so I will have to let everybody come in slowly. So just bear with me. I'll go through the list.
Good, but you might already start putting some questions into the chat because I can respond to these. Perhaps you can, in this time, say a little bit more about this last perspective. So how do you see this simulation part actually moving from AI? Can we, do we need different type of information to create now these models, or can we do this out of the same data sets which we use to create the hypothesis?
Yeah, that's a good question. And the answer is we can use the data, you know, the data that we get from things like WGCNA can build the skeleton of the model. It can give us an outline of what is interacting with other things or candidates. But what it can't do is give us the parameters. It can't tell us, well, you know, this is going to be fast binding and, you know, irreversible and cause this kind of dynamic versus the other. So there are some things that you can understand, you can predict about a system just based on the topology of the network. But in order to really get a true simulation and start running the kind of in silico experiments that we need, you have to really parameterize all of those interactions. And that requires a different type of data than the high throughput data that we've been working through. That having been said, it doesn't actually require a lot of data. One of the things you realize is that just looking at the network topology, some things are critical to a system and some things aren't. So it can help focus the kind of data that we need to build better models, ideally.
Yeah. Okay, I mean, I like that you started our series here, especially because there's always the concern that you're juggling data, squeezing something out of them, torture the data until they confess, yeah? But that you show that you can actually move to mechanism and hypothesis from these data with some pretty straightforward analysis from omics data to some things which make sense and then give you testable hypothesis.
Yeah.
Something which is should hold for in more general terms, that people should think about how can they use AI not to stop short at let's say getting an answer, but trying to convert this answer into something which is possibly new science.
Yeah, I think that one of the things that kind of surprises me is that in bioinformatics, people are usually just happy when they get a network and they call it a day. But like you said, you don't know, that network tells you nothing aside from the fact that there's correlations in your data set. And it's very easy to be misled by this, impressive looking interaction diagram and thinking, oh, that's what genes do. But it's not. it's an abstraction you've imposed on it. We've gotten around that right now by always checking for the correlations that we find in our data sets in an orthogonal data set, something that's totally different. Or alternatively, we try to kind of back it up with text mining so we know it's just not, an accident of our data. But ideally, we would be able to kind of build a system where we could make testable predictions. And that's ultimately a much more solid way to kind of verify your hypothesis.
Thanks. So there's now more and more people joining in who can also now unmute themselves or show their videos if they like to. So I'm opening it up for questions. Get us started.
Thank you, Alex. It was a great talk. So as you mentioned, finding good data sets is important. So could you share some of the things you would look for in a good data set?
Yeah, it's always bigger than most bench scientists think it needs to be. So typically, if you're used to looking, most of us in toxicology are used to looking at, a data set of, 20 chips is considered a large data set. But the answer is that's barely enough to even justify using an algorithm. So if you want to be able to use these approaches, you have to be realistic. That's not something that's going to be done in-house by one individual. It's something that has to be done by, similar to the TCGA, sort of a consortium. What I would like to see would be something similar to TCGA, but toxicology focused, so that we could really get enough data that we could, that would keep all of us toxicologically focused bioinformaticians, happy for years. Because at the end of the day, it's actually not that expensive. It's expensive, when you compare it to one study, but it's not that expensive when you compare it to the number of papers it produces. And it also has the added benefit of bringing down the cost of the high throughput study. So TCGA actually did reduce the cost of sequencing quite dramatically, just because they had to develop cheaper, more high throughput technologies. So it has kind of an added benefit of making the larger data sets cheaper in and of themselves.
Are these transcriptomics data sufficiently, let's say, standardized to really pool data and quality control them? And how does it look for other omics technology? You restricted yourself here to transcriptomics. Yeah?
I did, yeah. TCGA actually does have other high throughput data. It has methylation, copy number alteration, as well as a very basic protein array. So the protein array is more limited. I think it's 500 proteins, and it's an antibody-based approach. And I think that just goes to the fact that proteomics just remains kind of recalcitrant to these high throughput approaches that are highly standardized, unless you reduce the dimensions quite spectacularly. In the case of transcriptomics, the TCGA was done at a very high quality control level. So, so there are batch effects based on, where, in some instances the geographical location, but they're kind of easy to get rid of because you have enough data. Again, with enough data, it's actually easier to get rid of batch effects than it is with a smaller data set. So it for a bioinformatics perspective, it can't be underestimated how important it is to have large scale data that focuses on one specific problem rather than 10 studies with 10 each that are looking at smaller problems.
Good. Anybody else?
So I have a question about quality. Hello, everybody. So do you think, Alex, you propose potentially putting together a database similar to the TCGA for toxicological endpoints? Do you think that the quality of that data set would be better, worse, or comparable given the multiple hurdles we've observed with animal studies and toxicological analysis? Sorry, that's kind of a loaded question.
I got to be honest. I think when I first started this, one of my plans was to use text mining to start fishing through the data. But my suspicion is that the biomedical literature is so inflated with difficult to reproduce results. I'll put it that way. That like I said, the data is worth less than the sum of its parts, not more. In the case of transcriptomics data, the example that we used to look at estrogen and bisphenol A was large. I think it had 62 data points for each chemical, which is very large by a toxicology standard, but tiny from a big data standard. But that having been said, it was enough that you could fish out a lot of the data that was important to BPA and to kind of I don't want to say definitively, but kind of start pointing in a direction that is more useful than continuing to look at the same receptors and the same mechanisms that we had always assumed from the get-go that just turned out to be wrong.
Thanks.
Alex, since this is let's say an interest group within the department to further the use of AI and big data analysis in our department, what are things people could come to you? What would you be interested to work on? Where could you help with your specific skills and tools? So what are the type of things you would be inviting?
So it pains me immensely when people have a transcriptomic data set and they just run it through a pathway-based approach and they call it a day. Because in the kindest interpretation, you're leaving, some 60 to 70% of your data on the table. You're not even touching it. And trying to kind of figure out what the dark matter of your data is doing is actually not that hard from a bioinformatics perspective. It just takes a little bit of a different approach than most people are used to. So if you have data and you're just using overrepresentation analysis or any other pathway-based approach, I'm here to tell you that there are better solutions available, and I'm happy to help.
Okay, but you also understood you don't come with 20 gene chips. You want a little bit more to put your teeth into it.
Yes, but I'll still be nice if you have a small data set. I will do my best.
Okay, any other questions?
I do have one. So Alex, your talk was really good. So it is common to hear that it is not a good practice to derive a hypothesis from data. However, your argument makes a lot of sense. And so how are you dealing or how will you deal with resistance from when you publish?
That's a good question. I think the suspicion of hypothesis-free data is not misplaced, because it is true that if you have a large data set, any idiot can fish something out. And some idiot fishing something out that's wrong is problematic. And I think we've certainly seen that with the COVID literature. There's been a lot of things that have been published quickly based on big data that just turned out to be wrong for a multitude of reasons. That having been said, I actually think that a lot of the issues that we've been facing in science can actually be dealt with better by taking this sort of data first approach. So it's not, any accident that there is a reproducibility crisis in science, that a lot of the studies, this has been shown in a lot of different ways, whether it's the animal studies that we looked at in ECHA, whether it is, people trying to reproduce some of what's considered canonical data, for a lot of different endpoints. The level of reproducibility is really shameful. And the last thing we need as scientists right now is to give anybody a reason to doubt science. And the advantage of something like TCGA is that the data is there, it's publicly available, everybody can work on it, and you can do your analysis and publish your results. And if you're wrong, somebody will catch you very quickly because they have access to the same data. And you did see that with COVID. One of the big data, studies was caught within Twitter within 15 minutes just because of data quality issues. So I do think that, that yes, there are hazards with, just large scale hypothesis-free data collecting, but I actually think that those hazards at this point are better than the hazards that come from just continuing to do what we do with all the issues with reproducibility and publication bias and, just the fact that a lot of the data is not up to a high quality. Do your data once, do it big scale, do it correct, and make it publicly available.
Thank you.
I have a question, technical question. So now more and more we're moving towards single cell sequencing, and now you will be more and more seeing the data sets on the single cell sequencing rather than the transcriptomics from the entire tissue or whatever. So how applicable the tools you are using will be to single cell sequencing, and how much more difficult it will become to interpret or not more difficult? And another question is, do you still need more than 100 array sequencing data, taking into account that you from only one sample you will get much more data information than if you would just do normal transcriptomics?
That's a good question, and I haven't actually worked with single cell RNA-seq, but extrapolating from some of the problems I've seen in other high throughput data sets, I would say that the first mistake most people make is kind of assuming, well, if this workflow worked for, bulk cell RNA-seq, surely it'll work for single cell RNA-seq. And that's almost certainly not true because it has its own weird biases or, data quality issues that will cause problems for workflows that have been, for over, at this point, 20 years, optimized for basic transcriptomics. So I would be extremely, the easiest thing to do is just take, okay, I'll take WGCNA and apply it here, but I would be very cautious about doing that. The other thing that I think we're going to start discovering is that we've been thinking, well, gene Y has function X, right? But gene Y probably does not have function X in every single tissue in every single context. And you can certainly see that at a very basic level when you're looking at TCGA data, because there's about 1,000 genes or so that are prognostic for poor survival in one cancer and prognostic for good survival in another cancer. So it's either, somehow it's doing something very different or playing a different role. And as we start to get more granular data about what different genes are doing in different contexts and different cells, we're going to have to stop thinking of, oh, this is a cell cycle gene, or this is, an immune gene, and start thinking more carefully about not just what that gene is doing, but what that splice variant is doing in that particular time. And that's why I think ultimately the modeling is kind of key, because it gets at that actual functional interaction and what it's actually doing within that context a little bit better than just saying, oh, look at this network, look at this cluster of cell cycle genes. So I don't know how we're going to get there with single cell seq, but I know that we have to think differently about it.
Okay, cool. Good. Any other question?